Good afternoon. I guess we might as well get started. My name is Sean. I'm a staff scientist. I work for Peter Bannattini, and been here, I guess, from the beginning as far as the functional MRI facility is concerned. This is sort of a bit of an oddball lecture. I've been doing it for a little while. It's basically I try to describe a little bit of the history and the facilities of at the NIH that are dedicated to MRI and functional MRI, and give you some context for where the scanners that you use come from, but also some history of how the institution came about, and then also a little bit of what I call a postcard tour of the researchers here. So it's basically a little postcard for each researcher. So feel free to stop me and ask me any questions. And let me just start off by asking you quickly, show of hands, how many people here have actually run the scanner or are currently doing fMRI experiments? Okay, it's about 3/4 of you. And how many of you have actually been in the scanner yourself? Okay, it's a good idea if you ever have a chance to get in there, really understand what's going on. So that's basically a quick outline. I'm going to just tell you a little bit about the neuroimaging resources here, a brief history of MRI, and from the beginning actually, and some overview of the research that is being done across the facility, and then, as I say, these postcard presentations of work on the functional MRI facility PIs. I'll talk a little bit about 7T. I know you just had a lecture on high resolution scanning, but I get excited about the 7T, and so I'll sort of focus a little bit on people who are using the 7T in my discussion. Okay. So I guess one thing to point out is that it may not seem that way, but this place is actually one of the largest concentrations of MR research resources of just about anywhere in the world. It's a very deep, but it's also a very diffuse community. There are people here who have developed things like diffusion tensor imaging that you take for granted, people who were at the founding of functional MRI that we all take for granted, people who developed methods like arterial spin labeling that probably not that many of you use, but is also sort of an important functional MRI technique. People who've designed pulse sequences, people who've designed novel hardware, especially at the ultra-high field level. And you may be surprised to find out that a lot of people who have gone elsewhere started off their careers here like 20, 30 years ago. To start off with, a very short history of functional MRI. About 30 years ago, the building that most of the scanners are in was open. It was the brainchild of this guy called Ted Becker, who I think functioned somewhat as the intramural research director back then. And it was a little crazy because it was the first time the NIH the institutes really used to be very, very independent. And it was the first time they actually created a dedicated research facility that spanned all the different institutes. And even crazier was the fact that they devoted it to a single imaging modality. And they let it be run by physicists, which I think was probably the craziest thing, because what that meant was that the physicists, rather than being constrained by radiologists, and if you're a radiologist, don't take offense, but it meant you could do a little more crazy things than you would if you were run by radiology. The psychologists could run a little bit amok, and they could all play together in a way that I think actually led to a lot of really cool work. And in many ways, if you look at gold medal winners, it proved its worth. People like Bob Balaban developed magnetization transfer here. Alan Kretsky, well, he developed ASL when he was in Pennsylvania, but he came down here, not Pennsylvania, but at Carnegie Mellon. Peter Bazard developed diffusion tensor imaging, at least the mathematics about it, and Carlo Pioli made that possible for people to actually do those studies in humans. And there were some really entertaining people here as well who ended up, Bob Turner, who is kind of this, I'm not quite sure how to describe him, but a very central character in the development of EPI, and very central character actually here at the NIH as well, since he pioneered the first studies in humans. Denis Lee-Buhon was also someone who started his career off here in MRI and then went off back to France. and worked very hard in diffusion-weighted imaging. And I think, well, I'll talk about some of the other work that got done here, but pioneering work in longitudinal studies, both in multiple sclerosis and also in child brain development, which I think is actually one of the major contributions that we've made scientifically here. So NMR Center opened actually 30 years ago this fall. Ted Becker hired this guy called Dave Holt, who hired Bob Turner, and diffusion-weighted EPI got started on this EPI scanner. And I'll just tell you, one of the amazing things for me is that from the time that they decided that they were going to open a center to when they built the building and installed the scanners was 2 years, which, if you know how things move around here, is kind of like just a remarkable thing that they could do that. 1989, Harold McFarland, who just retired, started the first longitudinal multiple sclerosis protocol, and that protocol is still collecting data today, and they are still bringing back subjects who were scanned back in the 90s on that study, if they're the surviving ones. So just a couple of pictures from back then. So this is the original 4 Tesla scanner that we had that was basically across the way from where our 7T is now, and the person who is putting subject in, that's Peter Jezzard, who now runs the physics group at Oxford. And this is a patent application that Bob Turner made back then for a gradient coil, basically a local head gradient coil for that scanner. And this was what was used for the original functional MRI studies that were done here back in 1992. So I don't want to go into too much more depth, but I will just say that this paper that Bob wrote for the 2011 20 Years of fMRI special issue of Neuroimage that Peter put together is just a remarkable thing to read. He is a very good writer. He is not shy about his contributions, and he tells some amazing stories. So if you really want to get a picture of what things were like back here, and especially when things were really starting to get going with functional MRI, I cannot recommend reading that paper. I mean, it really, you will laugh. It's incredibly revealing and very fun to read. So that's one recommendation I'll make. So back then, Judy Rappaport and Jay Geed began scanning, I guess in the early 90s, on the 1.5T. It was an unshielded 1.5T that was installed by GE. And they just started scanning normal kids. And they weren't quite sure what they were going to do with that data. But 25 years later, some of the kids who were being scanned back in 91, 92 are being scanned again now. And I'll talk a little bit about that research later, but that was actually, I think, another one of the major threads of work that was done here. And you'll hear about that later, but these longitudinal MRI studies of brain development. Peter Bazzer published his first DTI paper in 92, and that's also the same year that they conducted their first successful fMRI at NIH, but I don't think they got on the bandwagon that Peter and Ogawa and Ken Kwong did. They managed to publish those papers in 1992. fMRI really started in earnest here in 1995, I guess, with Leslie Ungerleider, who started looking at motor plasticity with Avi Carney, came over from Israel, and they were doing the first of those studies in the motor cortex on the 4 Tesla scanner. And I should just point out that this is one of those great things about being here, is that there are a lot of resources. I mean, we all are familiar with the bureaucracy here, but the amount of resources that you can have access to is quite amazing. So I think Bob Balaban actually got an act of Congress to pay for the 4 Tesla scanner. And at the time, it was like an astonishing amount of money for a scanner, and it put us in the lead for a little while in terms of high field imaging. So, so after 95, things really started to take off. The Laboratory of Brain and Cognition came together. Leslie Ungerleider, Jim Haxby, Alex Martin formed this group that was, I think, probably the first lab that was focused so much on looking at using brain imaging as a tool to look at normal cognition. And in '99, Alan Koretsky was hired to run the NMR facility, which is kind of the, how would I describe it? It's basically our host that the functional MRI facility lives in. They're responsible for providing all the infrastructure, networking, and basically also the organizational world that we live in terms of human subject guidelines and so on. That was the same year that Peter Bannattini was hired to run this new functional MRI facility. And again, the functional MRI facility was something unique. It was basically a collaboration between mental health and neurology. They decided there was such a demand for functional MRI that they should actually devote the resources to build a facility dedicated to that, which again was considered a little crazy back then. And it's kind of interesting to think that at the time it was a very controversial decision for us to get a 3T scanner, because there were a lot of people who said, why don't we buy two 1.5T scanners? Why would you spend the money and buy one 3T scanner? I think in the end that was a wise decision, but there was actually apparently quite an argument at the time, because you had a lot of people who were quite happy at 1.5T, and why should you ever move away from 1.5T? I think we benefited from the sort of technical foresight from that. And then basically in 2000 is when the functional MRI facility really sort of took off. The program in NIH really started to explode then. I guess the functional MRI facility had been set up with the idea that there would be a lot of new investigators arriving who wanted to use functional MRI. And there's a guy named Dennis Charney who now runs the Mount Sinai School of Medicine, I believe. But at the time I believe he was running a committee that was looking at the future of the intramural program for mental health, and they decided they needed to have a program in mood and disorders, and so he became the program director of the mood and disorders program, and he hired a raft of investigators who are still here now. Things started to explode then, so we bought another 3T scanner that went into the bay where we now have our 7T. And our original 7T, which was unshielded, also got purchased and put in where our 0T 11.7T scanner is at the moment. But that was one of the first, I think it was one of the first three or four commercial 7T scanners. And for reasons that are a little unclear, it took us a ridiculous amount of time to get approval to scan humans in our 3T, the second 3T. And so Jeff Dunn and Peter had a collaboration, and they decided that they would use that time to develop something interesting technically. And they basically put together one of the first multi-coil parallel receive systems using off-the-shelf parts. And that actually was, I think, one of the first demonstrations of the utility of doing parallel receive on a 3T scanner. And the vendors sort of followed afterwards. But that was kind of, I think, a pretty important accomplishment. So then things continued to improve. There was a program here in the genetics of genes, cognition, and psychosis, and they were expanding rapidly, and they needed more scan time, and we needed a new scanner. So we decided we had purchased a scanner, but we had done this unique thing where we had purchased a scanner, but we didn't actually know where we were going to put it. And this caused us a lot of grief, and at a certain point we realized we were about to lose several $1 million if we didn't find a place to put it. So long story short, the area that people who use this facility know as 3TA and 3TB used to be one scanner, and we, after a lot of work with some architects and, well, mostly a really neat guy called Scott Grieve, we split that room up into two, and we put in two scanners where we had one, so we didn't lose the money, and you now have 3TA and 3TB. And around that same time, we inherited another scanner, which is 3TC, and then 2011, the facility pursued. Well, actually, it was a joint program between mental health and neurology. We decided that we were going to pursue a program in ultra-high field. So they Alan Koretsky, I guess, spearheaded this, and he basically got funding for us to buy the first self-shielded 7T. And I should just explain to you that up until when the 7T was installed, every 7T in the world that scanned humans had been unshielded. And that meant it was very, very hard to sight. The fringe field was massive. You had to put in huge amounts of iron, like several 100,000 pounds or tons, thousands of tons. And so we actually pioneered the first self-shielded 7T. We basically told the manufacturers we were not willing to sight in the traditional 7T area. We wanted them to build a 7T that had that was self-shielded, and we put it out for bid, and we actually started the whole 7T market in terms of making systems available that could fit in an old 3T bay. So we actually put that in one of our 3T bays. And around the same time, this was the first Siemens scanner that we'd had, was the Siemens 7T, and we replaced one of our 1.5Ts with a Siemens 3T, and that was an interesting discussion to go through since we had all this focus on longitudinal studies and people have been using this 1.5T forever for their longitudinal scans. And 2012 was kind of a sad year because we got our first 11.7, got to field, and then due to some unfortunate technical testing, we quenched it, and it got damaged and it got broke, and we had to send it back to England, and they couldn't fix it in England. They fixed it and they broke it, and now it's gone to Italy, and we're hoping that it's going to get fixed and brought back in 2018. So it's been doing a European tour for a little while now. 2015, we got our first Siemens Prisma, which is over mental health and neurology have about 50% of the time on that system. And then just this year, we've upgraded a 3TA to 3TB to the latest model of the GE scanner. 2018, we're going to get our second 7T. It's going to be the new model, FDA approved Siemens, pardon me. And we're also hoping that it'll also be the year that the 11.7T comes back and we are able to start scanning with that. And that would be one of the first, possibly the first operational 11.7s if everything goes right. So I think most of you know the lay of the land here, but just in case you don't, this is our 2 3Ts. This used to be one scan room. It was a really, really nice scan room. And our 7Ts down here, 3TCs here. This used to be the 7Ts, now the 11.7T, but right now it's just a storage area, basically. It got shipped back to, as I say, over to Europe, and we're hoping that it'll get reoccupied. The Prisma is over in this corner. This is the loading dock. And 3TD is here. That's the Skyra, and this is where the 7T will be located. There's the whole animal facility over here, mouse imaging facility, which we can talk about if we want to. Okay. So now that's enough of the history. So what are we actually doing now? There are actually about the scale of the functional MRI facility, I think, is a little astonishing. We have 70, I believe actually closer to 80 active protocols now that use our facility. And it's one of the reasons it can be so hard to figure out what's going on there. We have 30 plus investigators, PIs, and we scan about 6,000, we do about 6,000 to 7,000 exams per year that are mostly functional MRI. A lot of structural as well, and most of our clinical studies tend to be MS. It's, as you know, I think Peter will talk about this at some other point, but there's just a lot going on there, and there are studies in all these different areas: epilepsy, visual processing, mood disorders, motor function, auditory, attention, language, you name it. There's a lot. At some point, I thought it'd be interesting to sort of see how many papers have been published from from our facility and from the NIH in Bethesda from 1981, and right now it's about, I think we're now over 8,000 And that's a little bit too much to sort of comprehend. That's just MRI. So when we do a more selective literature search, we see that we've actually got more than 1,000 papers that have been published using the facilities of the functional MRI facility. But 75% from NIMH, about 20% from NINDS, and about 5 to 10% from other institutes. We have over 90,000 citations and an H-index, which is pretty large. And we go through this exercise each year of trying to figure out exactly all the papers that come out that are actually used in our facility. So it's about 100 per year right now, so you'd have to read 2 papers a week if you wanted to keep up with everything that was coming out of the functional MRI facility. And I don't think I need to go through this again. That's just sort of a timeline of when we've gone from 1 to 5 scanners. So we've got these 3 scanners are now identical. These photographs are a bit out of date, but they're all the MR750, which is the modern GE platform. 3TD is the Skyra, which is a wide body, wide bore, wide body. And then we have our 7T, and then we also have use of the Prisma, which is really nice, especially for people who are doing diffusion imaging. It's really sort of a design that's come out of the Human Connectome Project work. And I think nowadays most people are using 32-channel head coils. There's a few folks who are continuing longitudinal studies on 3TA who are continuing to use an 8-channel coil. But most people are using 32-channel coils on most of these systems unless they have particular reasons wanting to do, for instance, multimodal studies with EEG when you may need a bit more room. So this is a news clipping from when the NMR Center opened, and as I said, it was 30 years ago. I'm sure we should be celebrating it, but I'm pretty sure the person who would have organized it has just retired. Joe Frank, this is the opening. This is at the console of a GE scanner, and Joe Frank is still here, and I believe that Judy Ireland is still here a couple of days a week. Other folks have gone on to Manitoba, and some folks I don't really recognize, but it was a big day back then. And the reason to mention this is shortly after opening, this is when Judy Rappaport, and this is where I'm starting my postcard summaries of people's research, instigated a series of imaging studies looking at bringing kids back on a regular basis. They would scan every Tuesday evening. She hired a staff clinician named Jay Geed, who was here for quite some time, and just a couple years ago he left to go to San Diego. I think this is actually one of the major, if you had to talk about major research themes here, it's been these studies of longitudinal development. And they show this sort of famous change in gray matter density. Sorry if I seem to have lost my cursor. With increasing age, so what they call the pruning of the cortex, I think they refer to it as. This is the work that I think that is probably that they're best known for. They also did some work contrasting the development in childhood onset schizophrenia and compared that to normal brain development. So this is sort of really unique work that continues to this day. As I say, that in fact, Armin Rosnahan just became a investigator a couple years ago. He was working with Judy, and he is now bringing these people back who were scanned back in the 90s and the 2000s in what he calls the Bring Back Protocol. But he is someone who is also really doing exquisite work linking genetics and the environment to brain development. Very careful methodology. The papers of his that I sort of tend to read are the ones where he basically shows you how much motion in the magnet screws up your measurements and changes your estimates of cortical thickness, which is a real problem, especially with these studies in children. Another person who's come out of the Rappaport lab is Phil Shaw, and he is moved actually into the Human Genome Research Institute but he became very well known for looking at distinguishing the brain development between normal brains and people with superior IQs. And if your brain develops this way, I think this means you're going to have a higher IQ than the rest of us. He just does tremendous work. He's here just about every Friday afternoon, scanning kids on 3TB now, 3TA, I guess. And he's moved over from looking just at IQ, but also looking now at ADHD and looking at brain development in these children, and has been just does very careful longitudinal studies. So this is, as I say, one of these really neat themes that have come out of the research here. Moving over to someone who's more established, Karen Berman now runs the Cognitive Brain Disorders branch. She has been here for a good long time. She started off doing positron emission tomography, and she has a big interest at looking at, I guess, one of the pioneers of what you call neuroimaging genetics, and specialized also in looking at children who have something called Williams syndrome. Almost all of her studies are longitudinal, and she has been also looking at the effects of puberty on brain development, and just a real pioneer in that. She does all sorts of different types of imaging as well as MRI, and uses eye tracking, and has a large group working for her doing this multimodal imaging. This is just one of her imaging genetics review papers, looking at the difference between people with the Williams syndrome and normal controls, especially how they react to different types of fearful scenes. So one of the major technical pioneers here is Jeff Dunn. He's been here for, I guess, since the early 90s. I think the first paper that I could find from him at the NIH was in about 1990, 1991. He became an investigator around 2000, 2001, and he's been on the cutting edge of all sorts of different types of fMRI methods, especially at ultra-high field. He was one of the first people to look at how you could use phase in the images to improve contrast at ultra-high field. I think is well known for developing susceptibility-weighted imaging. And he's also, I think I mentioned earlier, the development of this multi-channel coil. So he has a program that is, unlike most folks here, he's got a very active program looking at sleep. So he's got probably the most intensive sleep studies going on anywhere, where he has people spending the entire night in the NMR 3T. He's got a real interest in sort of understanding contrast mechanisms and what the foundation of the resting state signal is. He also has a group working on hardware development. So in addition to developing things like the 16-channel coil, which I'm showing here, now he has a group developing methods for completely new types of transceivers with on-coil transmitters. And Jeff is the PI behind the 11.7 Tesla project. So he is getting ready for the return of that system and developing new technologies for that. So he's probably one of the most intensive hardware developers on campus right now. And then, of course, he's got real interest in understanding what the sources of contrast there are in susceptibility-weighted imaging, how myelin contributes to T1, whether or not you can actually really measure T1. So just a fantastic resource. And sorry, this is, I should say, this is actually this first 16-channel system that was built, for the 3T. It was home built, and it actually basically became the basis of one of the coils from a company called Nova Medical. But really showed exceptional promise for doing high resolution work, which I'll show a little bit later. Also in NINDS is Mark Hallett, who is one of the, I did my PhD in Montreal at a neurological institute, and whenever I mentioned anybody to there about coming down to the NIH, they immediately mentioned Mark's name, since he's so well known in the area of motor systems. And he has been looking at, he has a very creative group. They've been using neurofeedback. They've been evaluating motor disorders. They also do a lot of simultaneous EEG and fMRI in the magnet. I would say they're not necessarily the most, they're more applications than technical development, but they're always sort of on the cutting edge. And he has a staff scientist, Sylvina Horowitz, who's just absolutely first class fMRI scientist. Another sort of original pioneer in the area of neuroimaging is Leslie Ungerleider, whose name I mentioned before, and I think she was one of the early adapters, and I think arguably the user who probably did the most to stimulate the development of functional MRI here. I think she was responsible for recruiting Peter Bannattini. I think she had something to do with the creation of the functional MRI facility. And in addition to being an early adopter and doing a lot of very important work looking at decision making as well as functional anatomy of face processing, and she's very famous for looking at the two different streams, the dorsal and ventral streams in visual cortex. One of the things that is unique about Leslie is that she's maintained a parallel imaging program in primates as well as humans. And there's very few people out there in the world who actually manage to do both those things and do them well. So she's been using the vertical primate scanner that's in this building, as well as doing some work using a horizontal scanner back in the day. And at the same time, she manages this sort of parallel human imaging program, and has been combining TMS and functional MRI, and recently has been using concurrent ultrasonic stimulation in the magnet as well with one of her postdocs. I think what's one of the first, there are very few groups who are trying to do that out in the world right now. So one of her key collaborators is Alex Martin, who is really well known for, he's been around doing studies of the representation of knowledge and category representation in the brain. Back in the day, he was doing a lot of application work, in sort of basic research, but more recently he's been applying this to autism spectrum disorders and looking at how the brain circuits are different between typically developing and autism spectrum disorder kids. And even more recently, he's moved into doing some ECOG work, which is quite challenging since there's not an active ECOG program here at well, I guess there is now a little bit. And he has also done a lot of collaborations with people looking at the ingestion of food. And one of the things that one of his works that I find the most stimulating is a project he did way back when. This is, first author was Mike Beauchamp, who's now running the imaging program down in Houston. They used that first parallel imaging coil and did something I think kind of neat. Mike is very interested in sensory integration, especially visual, auditory, sensory integration. And what he did was show that areas of the brain in the superior temporal sulcus that people had thought was multisensory, when you were looking at it with 3 or 4 millimeter voxels, when you actually examined these images, at higher resolution, at 1.5 millimeter, with a 3T, mind you, they were able to show that actually, rather than having these multimodal patches, they were actually distinct patches that were visual, auditory, and much fewer that were actually multisensory. And I think for me, this is sort of a classic example of the importance of doing high-resolution imaging. I'm not going to spend too much time talking about Peter. You're going to see a lot of him, but he is one of the better fMRI scientists known around. I'm not sure exactly how you would describe his research theme. I think it's sort of maximizing information from the fMRI signal is one way he described himself. He's also very interested in understanding contrast mechanisms. He's done pioneering work looking at information mapping, especially when Nico Kriegersport was here. He's one of the pioneers of doing decoding, and also understanding what the resting state fMRI mechanisms are. This is a study that Javier Gonzalez did with him. This was looking at doing 100 runs and showing that you did this massive averaging and showed that there was basically evidence for activation almost everywhere throughout the cortex. And I think you'll be hearing more about this later on in the lecture series. Yeah, I won't rest on that. That's some resting state correlation work that basically showing how much of the signal that people measure in resting state is actually due to breathing and to respiration. So this is, again, in terms of the breadth of the program here. Jun Shen runs something called the MR spectroscopy core, and they are active developing methods for looking at spectroscopy, doing proton spectroscopy, looking at glutamine, glutathione, and glutamate, as well as recently they've been developing techniques for doing C13 on the 7T. They are, if you have an interest in doing spectroscopy, they're the ones to talk to. And I think someone from his group will be talking later on in the course. So continuing the postcard tour, this is Danny Pine, who I think mentioned joined the NIH back in the early 2000s and has now got a large branch looking at developmental and effective neuroscience. He studies anxiety and does a lot of studies with adolescents, especially. They actually have one of the most efficient operations for scanning around here. And they have taskmasters. They're extremely rigorous in the way they collect their data. He works very closely with Ellen Liebenluft, who looks at brain mechanisms in childhood bipolar and also does adolescents with severe irritability. And I think she's very interested in making sure we distinguish between kids with bipolar disorder and kids who are just irritable. And they have a new person who's joined their group who I don't believe has started scanning yet, Argelis Stringaris, and who's in the same area. But this is part of this very active pediatric neuroimaging program here at the NIMH. I'm going to quickly brush over Catherine Bushnell, who was recruited from here and started the intramural program in this National Center for Complementary and Integrative Health. She's a pioneer in doing imaging studies of pain perception, and one of her new recruits was Lauren Atlas, who another pain researcher who's very interested in looking at placebo effects. She's very multimodal, doing galvanic skin response, doing eye tracking, and very active on the Skyra scanners. I have to tell you that delivering thermal pain is just got to be one of the most technically challenging and not fun things to do in the magnet, but these guys are doing a good job of that. I guess Eric Wasserman is here. He does some work on doing transcranial magnetic stimulation, and I realize that this actually stimulates me to remember that we now have a new program in neuromodulation led by Holly Lisenby that's going to be doing concurrent TMS stimulation, which leads me to mention that we are going to be purchasing a upgraded EEG system that will have a built-in transcranial direct current stimulator system as well. So if you're interested in high-density EEG and neuromodulation, life is going to get more fun for you here. Eric runs the behavioral neurology unit, and he's one of the pioneers of TMS and TDCS, and has also spent some time validating near-infrared spectroscopy with fMRI. Very interested in neuronal plasticity. So Carlos Zarate is he's one of the folks working. He actually was a staff clinician, I believe, who now runs the experimental therapeutics unit. They do multimodal studies, and their specialty is looking at fast-acting antidepressants. They've become very active users of the 7T scanner, both to do spectroscopy as well as high-resolution structural, and they also have some very challenging studies going on with people who are at risk for suicide. They've been very aggressive about expanding their use of the imaging facilities. They're also doing combined EEG and fMRI with the ketamine, a group of ketamine subjects. These are people who are hospitalized, who are here basically inpatients for quite some time, who are subject for treatment for they suffer from major mood disorders, and they're here to test out the possibility of using ketamine as a treatment for depression. So I'll just quickly mention that Carlos is using the 17, and it seemed like a good time to just mention one of the reasons I'm so enthusiastic about MRI right now is just the possibilities of doing 7T work. And this is our 7T, which is, as I mentioned, I think was the first actively shielded 7T, actually, yeah. And we have folks, we were doing sub-millimeter anatomical studies on that pretty routinely, and we have people who are now, 0.8 is a little bit much, although that's not isotropic, but Lorenzo, I think, told you about some of his studies, so he's doing 0.8 millimeter in plane, up to 1.6 millimeter isotropic, and some people even going as far as 2 millimeter for some multi-echo studies. This is our coil for that, and just brings us exquisite data. This is some data from Ed Silson and Chris Baker, just showing the distinction between the fusiform area and ventrotemporal cortex, just showing beautiful contrast between face and non-face stimulus. And these are unsmoothed time courses. And the nice thing about the 7T just gives you just amazing contrast to noise. And that means that Chris Baker, who has joined the LBC a good long time ago now, has moved a lot of his research over to the 7T. He is very interested in seeing perception and plasticity, and he uses a technique called population receptive field mapping to map out visual cortex. And he's also sort of well known for thinking about circularity and reproducibility in fMRI, and is part of the the laboratory brain and cognition as well. I'm going to skip over that. And yeah, I think you saw this data. This is some of the high-resolution fMRI data that Lorenzo has acquired, showing the advantages of looking at blood volume imaging, where you can separate out the different layers in motor cortex, which is conveniently very thick. And final user of the 7T I'll just mention is Danny Reich. There's a couple of different people doing multiple sclerosis imaging, and he has been, I guess, inherited the mantle of Henry McFarland. Danny has a PhD in biophysics, as well as he's a neurologist and a neuroradiologist. And he's been really doing just fantastic work, especially high-resolution imaging of MS plaques on the 7T. So starting off with this sort of straight, here's your standards 0.7 millimeter 7T MP2 range. And there you are at 500 microns. And now I think they're now acquiring these routinely. It takes a while to acquire it, but the data is looking very, very nice. And they are doing that to look at, there's a whole question about what happens with the vascular supply to plaques and what the relationship is between the central vein going into these plaques, and they're able to get just exquisite data from the 7T, where they get much better contrast at 7T compared to 3T here. And the other thing, I guess, that they find with the high resolution is they're able to locate plaques that would normally not be detected on 3T, which they think might actually explain a lot of the behavioral issues that come about with MS. He has a staff scientist, Pascal Satie, working with him, who's just been doing spectacular work, both at high field as well as developing new techniques for doing detecting plaques. They're running a multi-center trial across North America using some techniques they've developed on 3T. Okay, so this is some work from Sam Teresi and Christian Grillon. This is looking at using the 7T. Christian Grillon looks at fear and threat, neurobiology of fear and anxiety. These guys are also do some challenging work getting scanning done with getting the approval for doing electric shock in the magnet, and they've been very aggressive about using the 7T to look at very small structures in the habenula, and I think this is the BNST stands for bed nucleus of the stria terminalis, looking at data which I think would be, they're able to replicate findings that they have found in primate but have not been able to look at in humans. This is, I think, a resting state study of connectivity in the the subcortical structures involved with anxiety. Okay, and then finally, some of the most recent work on the 7T is this is Eli Merriam, who's been looking at doing spatial maps in the looking at basically what happens when you move from one orientation to another, showing that there's actually orientation maps across visual cortex that are not necessarily just the orientation columns, but different parts of visual cortex have a predisposition for different orientations. He's been doing some work at very high resolution. He's scanning routinely now at 0.9 millimeter on the 7T. All right, so so much for my postcard tour. I'll just put up this image. This is the 7T Terra that's going to replace the NMR F3T next year. And I think the future is bright for everybody here. I think it's a great time, especially if you're doing ultra-high field scanning. I have dreams that, this is one of the few images we acquired on our 11.7 before it died. So one day it'll come back again and we'll have a human brain, a living human brain in there as well. Just encourage you guys to look around. There's tons of resources here, and anything we can do to help you, just let us know. All right, thanks a lot. If you have any questions, feel free to ask. Oh, sorry, that was a Kiwi. That's 11.7.